All right, welcome back everybody. Today I will go over kind of my experience using Roary, which is a Pan Genome Pipeline or tool. I've been really busy over the last few days, like week, building a Shiny app to do copy number variation, analysis and stuff. And then Shiny is like in the browser, so you should be able to just go to a website and then you could run your analysis that way. There's still a lot to do for it though, and it's been, like I mentioned, taking up a lot of my time. I haven't really had a whole lot of time to explore some of these other tools, but this one seemed really interesting. But yeah, when I get my Shiny app done, maybe I'll present it with everybody, and hopefully it's useful to do copy number variation analyses. So what is Roary? Well, right in the name it says the Pan Genome Pipeline, but it's a little bit deeper than that. So Roary is a program that will analyze a bunch of input annotation files with the sequence at the end, which is of note, in order to determine what the core genome of that bacteria species is. So say you had a bunch of E. coli genomes, you could annotate those genomes using the Prokka tool. So these guys used Prokka, like we did when we built our bacterial genome. And then you can feed in the output from Prokka into Roary, and it will determine the core genome. So the core genome is basically all of the genes that make that particular species that species. So if we go back to E. coli example, what are all of the genes that are common in all of the E. coli that we can say this is what makes E. coli E. coli? On top of that, Pan Genome is including all of the genes that are in like all of the E. coli, for example. So if we had 100 E. coli gene or 100 E. coli genomes that were annotated, we could find maybe a really narrow number of genes that are common in all of the E. coli. That would be the core. And then we'd have all of these other genes that are in maybe one to a handful of the E. coli genomes, and those would be like accessory genes or genes in addition to the core that would make the E. coli, special. Something that I kind of want to note with this is that this is a tool to look at already established names. So if we go and download a bunch of E. coli genomes, we are looking at the genomes of bacteria that are already labeled. I wouldn't use this, for instance, if I had a new genome. I wouldn't use Roary to identify which group it belongs to. There's a lot more that would go into placing a particular bacteria or a new species or a subspecies into a species than just looking at the sequence. So there's things like metabolites and growing favoritism that would help us classify a new bacteria. So in this main page that I'll put in the description, there's a couple useful links. So this step-by-step tutorial is what I had followed, and I'll kind of show the steps that I used going through here, as well as details on how to install Roary. Something that I would like to note is that when installing Roary, and we'll see it later on when I show my example, using Conda install Roary did not work for me. I had to do something a little bit different. So let's jump right into what I had done. First, I wanted to look for assemblies for bacteria that I could use in the pipeline. So what I did was I went to NCBI and I selected assembly from the drop-down, and then here I typed in Pantoia and then agglomerins. So just the regular old name. We could do E. coli if we wanted to. There's just going to be an awful lot of genomes. In my case, what I did is I chose latest refseq and I unchecked latest. I don't know how much that really matters since this is just playing around, and then from there I selected the assembly levels, complete genome, chromosome, and then contig. I didn't pick scaffold because there's 36, I think, here. Yeah, and that would take 1.5x longer to run, and it already took a really long time to annotate all the genomes. And then from there, there's 65, so you can just click download assemblies, and you can choose RefSeq or GenBank. I chose RefSeq and GenBank, and then from here I also chose, I did the genomic FASTA as well as the genomic GFF. Later on I found out that the genomic GFF from here does not work, so that's why it took a long time. I had to go back through all the different genomes and re-annotate them using Prokka as pointed out here. So I kind of had to do the NCBI work again. And I think there's a lot of troubleshooting stuff at the bottom too. So if you have issues, they might be worth going down here and figuring out. Maybe they already answered your question about what is going on. Anyway, so let's go to a step-by-step tutorial. When I downloaded everything, NCBI, they give you these kinds of files, so they're tarred. And what you have to do is you have to expand the tar into all of the different genomes. So in this case, this is the genome file. Here's the GFF file, which I don't need anymore. But in the FASTA file, these are what we can actually feed into Prokka. So now that we have gotten to all of the genomes, what we can do is we can start setting up our environment. So first we'll create our environment. So Conda create name, and then we can call it Roary Genome Pan Genome. How about? All right, and we can press Y and enter, and it'll automatically go through and do that. Additionally, at the very end, we can do -Y, but we'll do Conda activate Pan Genome, copy it, paste. There we go. Once we're inside, we can install Prokka. So we do Conda install -C bioconda and then Prokka. And that's the tool that we'll use to annotate all of the genomes. And I won't go through and install now just because I've already done it. Not install, run Roary and Prokka again. Running Prokka when I was using my computer, it took 6 minutes per genome, and there's 65. So it took a really, really long time in order to run all of them. So it's 6.5 hours. But we'll skip over that. Okay, the installation of Prokka is done. Now we can go and start working on getting what I have here as a genome file. So this genome file just has all the different genomes minus the .fna at the very end. I gotta make this way smaller. There we go. So we'll generate this file. And to do that, we can use something that's or a tool called base name in the terminal. So base name, if we dash, let's try dash help. There we go. So here's the tool. What we'll use is -S. So we're going to feed in the suffix or the last part of the file that we actually want to cut off. So following that, base name -S, we want to cut off .fna. So if we go to here, refseq, and then FASTA, we can see our genome file .fna. We want to remove .fna, and we need to point base name to that folder. And that folder is an NCBI, and then mine is refseq, and FASTA, and then we want all the different files in here. So we can do star.fna. So if we do that, we can see it prints out all of our genomes without the suffix. So we actually can now pipe that into genome.txt. So if we run this, and then we do head of genome.txt, genomes, there we go. We have the first 10, I think it is, here, genomes without the .fna. Now we can pipe this into Prokka. So if we go and follow this guy, this tutorial here, we can see for Prokka, we'll need to keep things organized. So our output directory will give it the name of our genome. So each genome will have its own folder called whatever the genome name is. On top of that, we'll give it a locus tag, and the locus tag is going to have the genome name. That way later on when it's merged, we have an identifier. And then we will need to feed it in that FASTA. So this is why we chopped off .fna, because then we can use this name here and here. So we're not going to simplify it like they did here. We'll keep on the whole name. Okay, so just like before with the other SRA numbers, we can do cat and then genome.txt, and that'll print out all the genomes one line at a time. From there, we can pipe that to parallel. Love some parallel. And then we'll give it one job. So for me, Prokka took a lot of resource, like a lot of compute power, and it took a really long time. Each genome took 6 minutes, like I mentioned, so it takes a really long time. And then we can do Prokka. We need to feed in kingdom. Yes, kingdom. So go back, we can follow what is here. We'll tell it bacteria. And then we need CPUs, which they don't have, but I gave mine everything. And then outdir. I put mine into a subfolder. They just put theirs directly in the working directory. I put mine in a subdirectory. So for me, I did Prokka / and then Prokka _ and then that curly brackets to get whatever that genome name is coming in from cat. All right, and then the genus that we're working with, genus is Pantoia. And then locus tag is something else that is in this guy. So locus tag. And then we just feed it in the genome name again. So that's pretty easy. Curly brackets. And then we need to tell it what are we actually going to annotate. So it's going to be the last genome file, or the last part is the genome file. And for me, that is in ncbi-genomes, genomes, -2020-1127. So it was a couple days ago. And then refseq -fasta / and then curly brackets .fna. So basically we're rebuilding what we broke down with base name. That's just the last part. And then we can finish it off with a quote. And then if we ran this, it would go through and it would generate all of these folders. So each genome gets its own folder, and inside it has all the annotation information. So this actually is the GFF file, I believe. I have to rename them in order to run one of the end parts. So that's why this is, I just renamed this wrong, but I think this is the GFF file. All right, so say we ran this, doop, and now we have all of our annotations done for Prokka. What is the next step? Well, we need to install Roary. And like I was mentioning before, we have to install Roary a different way than what is explained in the installation, how to install. So we can see here, it has Conda install Roary. I don't know, there's not a link. Yeah. When I try to install this conda install-c bioconda Roary and try to run it, it doesn't work. However, if I go back and I do conda install-c and then bioconda label CF 2019-01, then Roary, it does work. So I would suggest running the second one if the first one doesn't work, or even just starting with the second one. The only problem is if you go and do like something like conda update all, it will change this back to bioconda, I believe. So once it's installed, I just left it go. But like I mentioned, to install, I did conda install-c and then bioconda /label/cf 2019-01 and then Roary. And that will install it along with all the dependencies needed in order to get Roary working. And we will wait for this to finish. Okay, now Roary is installed. We can go ahead and run it. So let's look at the help page, Roary -help. Okay, so the different flags that we'll use is -e. So we're going to create a multifacet alignment. So the output will basically have all the genomes, and then wherever there's a gap or something, it'll have like a dash. We'll use -n, so this will be mapped, and alternatively to using -e and then -n is you can do -e -maft, which I believe they have on here. Yep. So Roary -e -maft. Then we will do -v, so we want it to tell us everything that it's doing. So it's going to be -v. Then we'll do -p. We'll give it as many cores as like the computer that we have has. And then we need to feed it the location of all of our GFF files. So Prokka will, or not Prokka, Roary will use all the GFF files at one time. So we can just feed it kind of a path, a generic path to where it would go. So if we type that out, what does that look like? We'll do Roary, and then we'll do -f. I forgot about that one. That's going to be the output directory that we want everything to go to. And that's going to be the current folder. And then I put mine in Roary output. So we go back to mine, Roary output. That's where I put everything. Okay. Then we'll do -e like I mentioned, and then -n. So we're going to run and create that multifaceted alignment of the core genes, and then we're going to use maft. Then we'll do -v, so we want it to tell us everything that it's doing. -p, we want it to have the resources that we have available. And then we'll do Prokka. This is where we're feeding in our GFF3 file locations. And like I mentioned, mine, I renamed them wrong. So this is supposed to be .gff, or maybe, yeah, so I think this is the .gff one. But anyway, the file path to get there, so we need Prokka, and then we need to look in all of the current, or all of the folders in the Prokka folder. So the */*, it's going to look through every single one of these folders. Then we need /, so we need to go inside of those folders, and then *.gff. So we're going to look in the Prokka folder from where we are. So Roary Pan Genome, go into the Prokka folder, /*, go into all of these folders, and then *GFF, we need to look at whatever file has GFF at the end. Like this guy. Let's see if I open this up. Oh, it's a big file. Yeah, so this is the GFF file. GFF version 3. Great, so now if we were to run this, and I already ran it, I'm not going to rerun it just for the sake of time, we would get an output in Roary output. So if we go to Prokka, one more, go to Roary output. This is the output that we get from Roary. Let's go ahead and check this guy out. So we can see the core genes that Roary had found is 2922. So 99 to 100% of the genomes, somewhere in there, basically all the genomes, have 2,922 genes in common. So soft core genes, so if we expand the number of genes a little bit, so maybe not all of the genomes have them, we can add 630 to that. So the top 95, how does that go? 95 to 100% of the genomes would have 2922 plus 630. And as we go further and further out, we can see that less and less of the genomes are having more and more of the genes. We can visualize this a little bit better if we get a Python. So here is Roary underscore plots. If we go back to the step-by-step tutorial, in here they have the ability to plot. So Roary plots.py, I'll put a link to that down in the description to this page. And all you have to do is click raw, and then you can copy this, and then in this folder, in your main folder, you can do wget, right click. I need to copy again. Copy, paste, and then it downloads it and it puts it into the main folder. So this is the one that I downloaded before. If we go here, rename it. So we have Roary underscore plots.py. To run that, in order to make the pretty figures that they have shown on here, so we have the cluster, the number of genes by the genomes, and then this plot. So this is the one that's going to be more of a depiction of the text file that we just looked at. We can do Python. So we need to tell Bash what program we want to run. We want to run Python, or what kind of a script, and then we want to run Roary underscore plots.py. Now we need to tell it our NUIC file. So in the description on here, they must be using a different version of Roary, but ours is named a little bit different than this one. If we go into our Roary output, we can see that ours is called accessory binary genes.fa.nuic. So we can do Roary output, and then we want to tell it accessory binary genes.fa.nuic. All right, now we need to tell it the gene presence and absence. So that's going to give us, let's go back. That's going to give us the color. So presence, absence. And that file is called genes, presence, absence. So we do genes, we need to go into the folder, Roary output, genes, presence, absence.csv. Go ahead and run. Oh, I forgot. We have to install a few different Python packages. So we have to do Conda install. Let's clear this up. We need to do Conda install, and we need to do 3 of them. First one is going to be matplotlib. So that's what's actually going to help us generate some of the plots. Then we need Seaborn, as well as BioPython. Go ahead and run that. We can do H and then enter right off the bat. So whenever it gets done finding them, it'll go ahead and immediately install them. Great. Now we have our 3 packages installed. We can just be able to go up and rerun this guy. And what this is going to generate is the 3 plots that I kind of pointed at before. It's going to have Pangenome frequency, matrix, and pie plot. So if we open up Is that text file still open? Let's see, I think it's this guy. We'll compare this to the pie plot. All right, I need to get that back. Boom. All right. So our core genes, we have 29, 22. So all of our strains, basically all of our strains, our genomes that were input and annotated, have 2,922 genes in common. So those are the core genes that would make Pantowea agglomerans, Pantowea agglomerans. So soft core genes, so if we start broadening out the genes and dropping the number of genomes which they're involved or which they are included in, we can add 630 by going all the way down to 61 genomes. So at least 61 genomes would have 630 plus 2922, and so on and so forth all the way down. We can see out here, this cloud are basically the accessory genes. So 8 or less of the genomes would have these 13,228 genes. And if we go and we look at in Roary output, that gene presence absence, and we can scroll all the way down, we can start seeing here on the right hand side, let's go back up and lock our first row. Freeze panes, freeze the top row. Okay, the names are really hard to understand just because they're just in accession. But we can see here, this gene, there's 28 isolates or genomes that have it. And we can see right off at the start, there are 4 here that do not. And this one does not here. Then we get another one back, so on and so forth. So if we were to look at this as kind of a matrix, then we would have this guy. So where all the isolates are blue, those are our core genes. And as we start stepping further and further out, we get closer and closer to our accessory genes. These two here are going to share these genes, so on and so forth. So you can see blocks of genes for the isolates when they have those in common. And that's what this plot is kind of made out of. And like I mentioned, as we get closer and closer to the end, we can see that like these genes here are only going to belong to, I think it's this guy. And these genes here are only going to belong to this guy, if that makes sense. Yes. Go down to here, we can see that as we add more genomes, the total number of genes is going to increase. So when we look at our, let's look at this to make it easy. When we look at the total number of genes, we can see that there's a really high number. And the number of core genes is low. So as we add more and more genomes, there's an increasing chance that the core genes keeps dropping off, or we have a genome that doesn't have one of those core genes. If we're going to compare 2 genomes, a lot of them might be similar, but if we keep adding more and more and more, we keep increasing the chance that we have to drop some in order for all of the genes to be in all genomes. And as we drop more and more, then they become unique, and we end up with a really, really high number of total genes, as we can see here. Let's continue on. So, like I mentioned, the number of unique genes is going to increase as we add more and more genomes. So as we add a new genome, there's a chance that some of those genes are not in the previous ones, so we can increase. And then new genes, so new ones that are not in the previous total would continue to decrease to. I tried looking at the dot plot, but I couldn't find anything online. But if we look at the interactive tree of life, so this is ital.embl.de, so I think it's the Dutch website. We can see this is what the Newick tree that it's using to make this matrix. You can see that looks very similar to this, looks like. And we can just play around with some of the labeling, and you can export this and look at it someplace else too. But I think that's where I ended up. I haven't been, like I mentioned, I haven't had a whole lot of time to dive too far into this, but it is a really interesting tool, and I would like to spend some more time learning more about it. I'll put a link to the Roary plots.py in the description as well. I tried doing the DB generation that is in the step-by-step tutorial, and I couldn't get that to work in the little amount of time that I spent trying. This guy. It did create some files, but the locus sequence, there was nothing there. But yeah, if you guys found this helpful, like and subscribe. I'll keep trying to explore some of the different tools that are available out there. It is really, really kind of fun to play around with the different tools that maybe you had never used before, and you're able to see what's available. So yeah, thanks for watching, guys.